Um, so someone reminded me about the greedy thing that I had asked you guys to do. I had forgotten. But can you come up with counter example of a situation where you have denominations of coins or notes and you cannot return change? Let me phrase the problem again. So you have some denominations d1, d2 up to dk. And d1 has to be 1. Okay. The rest you can choose as you want. Okay. So that, let's say you have to return change rupees n. Okay. The way you do it in a greedy algorithm is remove the biggest denomination dk. This is in ascending order, right? This is the smallest one, this is the biggest denomination. And you take your change, the amount that you need to return, take the biggest number. If that number is less than n, you give one note. Okay, so now you're left with n minus dk. Now you look at this amount. If dk is still less than n minus dk, you continue to give these notes, right, until you hit a point where whatever you're left with is less than the dk, in which case you proceed to the next denomination and give coins or notes from that. Okay, so that is the greedy algorithm. Just take the best possible move at that time. So now can you give me some example? You can choose your k, you can choose your d2 through dk, okay, and give me an n where the method will fail. So it is, even though dk should, the greedy method says dk should be given first, when there is a possibility when n is bigger than dk, there is a situation where dk should not be given. You know, the 100 rupee note should not be given and something smaller should be given, because your goal is to give fewer number of notes. Okay. So anybody has done this? Yeah? No, not Indian denomination. Any arbitrary thing. You can have 22. Yes. Okay. 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 So you have to have one. Okay. Next is 15 and 25. Okay. So you have 3 and you're returning 30. Okay, so if you do the greedy method, you will have to give one note of 25, right? You're left with, so you give one note of 25, so you're left with 5. So you have to go, you can't give 15, you can't give 25, so you have to give 5 of ones, right? So you get -1, you do 5 times. So how many notes do you use? 6. And the best solution is, of course, to have 2 of D2, right? That would be a better way of doing it. And you can do this. How would you then solve this problem? How do you find that the answer to be 2 times 15? Sorry? But how do you just make that call? So, I mean, so right now you could see that 15, you could tell immediately that 15 times 2 was 30, right? And that is definitely better than giving 5 1 rupee notes, right? So instead of 1, yeah, so correct. So that if you had a 5 also, this this counter example will not work, because you can give a 25 and a 5 and you're okay. You're still optimal, right? So how would you solve this problem? greedy doesn't work, at least greedy in this way doesn't work. But there is still some substructure in there, right? You can think about it. It's not in, I'm not planning to talk about this anymore, but think about getting a dynamic programming approach to solving this problem. So you have an arbitrary set of denominations, d1 through dk, and you have an arbitrary number, n, that you need to return. The only thing you are guaranteed is that n is a whole number, right? And your d1 is 1, right? That you have to have. All right. So people asked me about which book I'm using and whether there's something I can recommend so that you can go through it better. So this is, now I've been giving wrong answers, so this is the correct answer. Okay, so this is the book that I have been, I used to use at some point. Okay, and that's a good book to use. It's a little, it's a small book, so it's fairly dense. So when you read it, you know, many steps are missing. So, but it's a good thing because then you can derive the steps and try and figure out what, how you get the next, how you get each derivation yourself. So. So yesterday we saw how to align sequences, and we took examples only of DNA. But technically you can do this, and this is used a lot for proteins as well. Okay, you just need to have a good sense of what is similar and what amino acids are similar and what amino acids are different from each other. Okay, so here you have a matrix. It's a symmetric matrix, so they've only put the lower matrix down here. Okay. Where you can see every point tells you how much to score, every cell tells you how much to score two amino acids when you see them in an alignment in the same column. And this is derived by looking at proteins that you know to be similar and then computing these scores from that fact, and also with some expert knowledge because you want hydrophilic amino acids to, you know, be similar, scored similarly, and so on. So this just all the things that we spoke about yesterday work also for proteins. You just have a very big alphabet size of 20 instead of 4. Okay, so going back to the sequence alignment problem, right? This is what we spoke about. I'm going to go a little slower so that you can actually do the calculations yourself. Okay. So we have two sequences, size m and n, okay, and you want to output finally a list of columns. Okay, each letter is either aligned with another letter from the other sequence or with a gap. Okay. And we said that instead of doing the whole problem in one shot, we are going to do all the prefixes. So you're doing the I prefix and the j prefix of x and y. And to store the solution, you're using a matrix called V, okay, and you score it this way. So the idea is that if you're extending an algorithm, a previous alignment by matching I with j over there, then the match score comes there, and you extend the previous alignment, so the previous alignment is there too. Okay, that is the Vi minus 1, j minus 1. Alternatively, you are aligning the jth letter with a gap. Okay, in which case you take the other, you go up in the cell. You take the cell from top. Okay, or you take the third option where you're aligning the other location with a gap. Okay, so we had done we had started off doing these two sequences. Okay, this is my X, AAAC, and my Y is ATC. Okay, so we have to fill up first the initial conditions, right? Which is this? Question anywhere? Did we do this? I think this was a G, but otherwise I think that these were the... Yeah, okay, sorry, but that's just one minor. Huh. Yes, it is the same thing, right? We did this exact same, very similar thing we did that. Yes. Okay, so when we are initializing this way. Correct? All your rows get to I times G and J times G. Okay, now we also need to remember, now this is a trivial remembering, here you have to, all this comes from extending a gap. Okay, so you do these arrows. Okay, that just tells you where to go next. Okay, it will become clear in a bit. Okay, so now let's say you're filling up this cell. Okay, the circled one. And let's say we are doing it row wise, which is what we did yesterday also. So now for the circled one, you have to solve these three expressions. Okay, so the first expression is 1 because A and A match. Okay, you remember the matrix, right? The scoring matrix. If the letter matches, then it's a 1. If it does not match, it's a -1. If it matches with if it is put with a gap, it's a -2. Right? So you get a 1 plus 0. Because 0 is where you are not aligned anything to anything. Okay? So you get a minimum is 1, so you fill it in. Right? Now you remember how you got there. To remember how you got there, you put that arrow. Okay, that says, what does this arrow say? There is no gap. You have to align me with whoever is on my left, with my I index and J index. That is what the arrow says. Okay, the next fellow, matching is a and T and an A match is a -1 plus I -1 J -1 is that -2 there, right? So what is this cell telling you really? What is the best alignment score when you are aligning just that A with AT? Okay, that's what it's telling you. Alright, so if we extend, if you're doing this, it means that we are matching A with a T and you're extending that one, right? You're extending a current alignment and matching A and T. Alternatively, you are going to extend a gap. Okay, so that's a -2 plus a I -1 comma J. Okay. So that the minimum is -1, and this time you remember you come from this left side node. Okay. So far, yes. Yes, correct. No, say that again. So shall I go back to the expression? Huh, okay. Correct.
So now the arrow from 1 to 0, that is 1 to 0, 0.
Yeah.
That includes that it depends on that 3 position.
Yeah. It depends on all 3. But which one did you choose? It always depends on all 3 because the minimum is taken on all 3. Okay, but the one that was the minimum was the first guy. So that is just to keep track of how you got there. Okay. Okay, let's do the third one. Again, the first one says you match A with a C, okay, and add the previous whatever alignment you had so far. What this really means is that these are both aligned with gaps, right? A and T are both aligned with gaps. That thing is saying that minus 4. Okay, so it's either minus 5 because C is different from A, okay, or it is minus 2 plus minus 6, okay, which means your A is aligned with a gap, okay, or this is a or it comes from there, which means your C is aligned with a gap, right? And so you get a minus 3, which is the minimum. Just telling you to go there. Mistake? Is the max? Did I say minimum? Sorry. I mean maximum is minus 3. Okay. So, what about the next one? What will it be? So you match A with an A, right, which is the first sign, okay, or you match A with a gap, right, and so on. So you get the best way to do it is what? There are two ways, right? You can either you can extend that, you can match A with the A, right, or you can match the first A with the A and the second A you can match with the gap, right? So these are the two possibilities. So you keep both of them for now. Okay. Now I'm going to do the rest of this. Okay. Just because because I have done it. Okay. So this is what the matrix looks like at the end of it. And the total cost of aligning the two sequences is out here. It's minus 1. Okay. So how do we get the sequence back? We have to follow the pointers. Okay. So the fact that the pointer goes up means that my C, the two I and J are aligning with each other. Okay, so I get a C aligned with a C. Okay, my top matrix is top sequence is AAAC, right? This is X, and my Y is ATC. Okay, because this is my I and that is my J. Okay, so my C gets aligned with a C. Now I'm in the minus 2 box over there. I'm here. So I can either go there, go here, or go there. Right? You can pick one. What would you like to pick? Up. How many want an up arrow? Okay. So then everybody else wants a diagonal arrow. Let's pick the diagonal arrow. So the diagonal means that you match A with the T. Huh? Because you are still going diagonally. So you have an A that you match with the T. Okay, you go up here now because you followed this arrow. Now from here you can go either here or there. Which one do you want to go? You want to go diagonal still? Okay, so let's go one more diagonal, which means your A matches with an A. This one, right? A matches with an A. So you have to go here. Now over here you have no choice but to go up. Okay, so that means this A is matching with a gap. So you have an A here that matches with the gap. That is your final alignment.
If in the last step, if you went upwards.
Yeah, let's do that. Okay. So you have, so far you have this, right? So over here you are saying let's go up, right? So if you're going up, that means your A is aligned with a gap. So that means your A here is aligned with a gap, which is, now you come here. From here you have only one choice, right? So you go diagonally, so your A gets aligned with an A. So both these alignments are optimal. In fact, there must be more, because we made a decision somewhere else also, right? We broke a tie arbitrarily here also, right? So we went up here, and from here we continued going diagonally. So we could have gone up, which would mean that this A, instead of being matched with a T, could have had a gap, right? So what happens there? So you have a CC that has to be matched, right? And over here your A is matched with a gap in this case. From here you go to a match. So this A now matches with the T. And then I guess you match A. This is also as good. This will all score, yes. This one, yeah, so you started with C, same, right? From here you have to go diagonally up. Whenever you go diagonally up, that means you took this option, which means you have to take the scoring matrix into consideration. So you got a C and a C, right? Now you're here. Over here, instead of going further, again you went an arrow, again went diagonally. Because you went diagonally, you are matching A with a T. Okay, so you matched it, went up. So you got a T. So far it's the same. Then from here you went up. You could have gone anywhere. Whenever you go up, that row is matched with a space. Because we are not done with the column here. We are still in that column. We are in that same column. We have not figured out where that fellow matches yet. But because we are no longer in this column, that means that we have put a gap for this one. Right? Come here. And from here, then you have no choice but to match the A. Yes? 2, 1. This. Yes. That means the A could not be aligned with an S. It came from here. It came with a gap. Right? It came from this situation. Because it came from this situation, we cannot align it with anything but the gap. The gap is what it's telling you to align it with. And we've still not done with J. That's why we are still in that column. Right? And then that is the one that we've not done yet. So this, how many of you like coding and would like to code this up? You're most welcome to do this now. Alternatively, you have to do it by hand, not this problem, slightly bigger problem. It's up to you. No. So filling up the matrix is fairly trivial. Okay, you just have to follow, just two for loops, no? For loop for the row and for loop for the column, or whichever way you want to fill it up. And the tricky part is getting back the alignment. Okay. So how many of you, I don't know if Sitabra did this. Did he do any algorithms on graphs? Like today he did he do depth first search? Breadth first search he did? He did a breadth first search. Okay. He did not. Okay. So one way you can figure out the alignment is to, if you just want one alignment, it's okay, right? It doesn't really matter. You just keep on following anyone. You can break the tie anyhow. Yes? Otherwise, you have to find all paths from here to any, to 0 actually, right? You have to find all, this is actually a graph problem now, right? These guys are your nodes. This one, these edges are your directed edges. They're unweighted. There's no weight on them. And you have to find all paths from here to there. And each kind of edge just means a different thing, whether you align with a gap, whether you align with a letter, or you align the other letter with a gap, right? That's all it means. Okay, so there are ways to do a search on the graph, okay, that tells you how to, that can help you enumerate all possible paths from this to there. Okay, a depth first search works well in this. Alright, so there are two different paths when you follow it. Okay, so here's one. Okay. And how do you follow the path? Right, so from here, this path is saying from here you go up. Okay, which means that your C, this C, you're done with your C, right? You're going up, that means you're done with your C in the row, should be matched with a gap. Okay, and then you go up here, right? Now you're here. Over here, you've taken the call to go diagonally, which means you have to match your G with this G. Okay, so you write that over there. Now when you're here, you have taken the call to go up, which means again your A does not have a partner, so it's going to be matched with a gap. You keep going up, okay, until here, after which you're matching the G and G, so you got the alignment. From here, you're again going further, you're matching the A and the A, and you keep doing that until you go to T and then A. So here you're matching the A with the C. Okay, so it's a mismatch. So the 0 minus 1 is how you came there. Okay, this is the other way of doing it. Okay, in which case you are actually matching, you're doing a mismatch over here with the C and the G, right? And so on. You keep going diagonally until you go up, so you have to introduce a gap. You go diagonally and then you go up, up, up. Oops. Yeah, so that was it. Okay. If I go back, I have to go through the animation again, so let's just leave it there. Okay, so there were two paths, and they both, if you look at actually the score, they will have to score the same, because unless our dynamic programming algorithm is wrong, okay, they have to score the same. Okay, so the time taken by hand is a lot, right? It is, I mean, it's the same number of steps, n into m, okay, for the computer. The space is also n into m the way we have done it, right? Because you have drawn the whole matrix, right? And you've kept it in memory. But you can compute the matrix just row by row, right? That is what you did. So you can, in principle, just keep 2 rows in memory at a time. Whenever you're done with one row, you just copy it on the first row and then keep going, right? At a time, you only need the row before you and the current row, right, as you're filling up. So you need only 2 rows. Or you can do it by column. You need 2 columns, right? But we cannot, how do we then trace back the pointers? For the pointers we had stored in every box, right, where we came from. So this, to answer this question, so why are we doing this? It's because your n and m is really big, okay, so you don't want to save all this in memory. Normally you have 2 long, big genomic sequences, so you don't want to do this in quadratic space. You know, as little space is better, is good, right? Because you have a million characters, so million times million becomes a lot to store in memory. Okay, so So an algorithm that does this in linear space does it in a divide and conquer kind of way. Okay, so we've done divide and conquer before when we did the binary search, right? We split it into two pieces. We checked which piece to search for the name when it's sorted, right? And the other example that we did was power, right? We could do the power by splitting it into two smaller equal problems. So here, the way they do this is take your sequence x and take the midpoint, okay, which is m by 2, okay, and see whether it, there are two cases. It either matches a space or it matches a character from y. Okay, there are only two options. Okay, so if we can find the if we just write optimal for x and y as the optimal answer, then just the same dynamic programming approach says that if it matches with a space, then you have to solve the left hand side and you have to solve the right hand side, or alternatively it matches with some character yj, in which case you solve the left hand side and you solve the right hand side. Okay, of course the catch is you don't know what the j is. Okay, but what this is saying is you're matching a prefix, arbitrary prefix, with a specific prefix of x, half the length of x. Okay, so we are actually calculating all these numbers. So what does each cell mean? So what does this cell mean? What does -4 mean? This is the maximum similarity score of aligning this prefix of x with this prefix of y, right? This is the complete answer. This here is prefix of x, prefix of y, right? The little thing at and the ctag. This is the answer for that. So when you guys calculated the first 4 rows, right, you had actually done half the problem. You had done it for the prefix. Okay, so when you do the whole row, what are you actually calculating? All prefixes of y you're matching with half of x. That is the answer. You did this. Okay, and this you know you can do in linear space. You do not care right now about the pointers. Right now you're just saying, can I find out all the possible prefix scores that's out here? Does that make sense? So you have this row, you can calculate from that row, right? And you keep doing this only row by row until you hit half, x m by 2, and then you stop. Okay, those numbers are all the scores of all the prefixes of y that match with half of x. Okay, that is right there, then you've done that. Okay, that was the left hand side of that expression. Those are all j's. Now the right hand side, let's go to the right side. This is the suffix of y, right, matching with again half of x, but the second half of x. Now we are not really computing this value, right? We are computing all the prefixes when we are constructing our alignment. But can we do that? How can we do this? You know how to do the prefixes. How can you do the suffix? Column wise, even if you do column wise, you will be doing prefix of all prefixes of x's with y. You're not looking at the second half of the sequence. This is the later part. You can reverse both the strings. That is one way of doing it, in which case this guy becomes a prefix. So you can do the whole exercise again, which is still only constant, right? Again is just 2 times. Or you can also just fill up your matrix from this direction. If you just take care into account, right? Instead of filling from up to down over here, you can fill from up on this corner to down there. That is a similar way of doing it. You'll have your equations will change a bit. Okay? So you can do that also in linear time, quadratic time, but linear space. And then you can find out what your j should be by adding those pieces and taking the minimum, whether it's a specific yj or a gap. You can do all those additions again in order n time. Okay, so what it ends up doing is it increases the time by a little bit by a constant factor, but the space is constant space. It is not quadratic. And let's see this one. So let's say you are able to do this, find out what your j is in mn time. Pencil. Okay. So then you have a t of m comma n is equal to constant times mn, right, to figure those out, plus you have this problem to solve and that problem to solve, right. So you have t of m by 2, that problem has become small by half, the x part, right, and we don't know what that second part is, depends on j. Okay, so that's it, we call it j plus t of m by 2 n minus j, right. Does that make sense? Because we can find out what this j is in quadratic time. Do you agree with that? We just found the prefix, all the possible prefixes, not the optimal, all possible values. We find all possible values here and then add them up with a gap, with the correct, if you're taking a j here, then from here you have to take j plus 1 onwards. Or you try it with all j's by matching here. This would be the score of matching plus the score of that plus score of this. All this you can do in order n time and mn to get the optimal, right? So you can do that part in some constant times mn time. And then you have to do the same operation again. So you understand, if you find the j, that's how you are figuring out the alignment. The answer you can find out in linear space, no problem. We are trying to get the pointers back, which we have lost in the process. So now you don't have to store any pointers in your matrix. All you're doing is figuring out at every point what j matches with the middle. And then you have two smaller problems. So this problem can again be, this time is again equal to, when you do it again further, it will be c times mj by 2, right, plus I'll do this one also, c times m of n minus j by 2, and two little pieces here and two little pieces here, right. When I'm doing this, I do this, I get this, and I have to do the smaller pieces recursively. And so you'll notice that, and something, okay, which I'm not writing, which is again going deeper. So you'll notice that this plus this is again constant times mn, right. Eventually this ends up being mn time if you solve this. Okay, so we are not really making the time complexity worse in the asymptotic sense. Okay, we are making it worse just by a constant factor. Okay.
How can we encounter the problem if m is odd?
M is odd. Then you take the floor. You always take the floor. Just decide. Approximately it will be half. Okay. All right. Let's do another small example. Okay. Let's say you're aligning these two sequences. Okay. This is too simple to ask you to do, so I will do it. So you get these two answers. Let's say there are many answers. Okay, you will notice there are many answers. You can put your gap anywhere, right? Just looking at you don't even need to do dynamic programming. You can just align it. So here's one way of doing it. Here's another way of doing it. Both are optimal. Correct? You agree? Because you have to match three of the t's with a gap, right? And the others, the A has to be matched, right? And the others can just, you can just choose your t's. Now, if you ask a biologist, she might prefer this one rather than that alignment. Would you agree? Because if you think about, if you remember the mutations, right, I spoke about earlier, yesterday, the way you have an insertion, you have a deletion, what this means that you have three different events. Okay, here you have just one event, because this is like saying that a chunk was deleted. Instead of saying a T here was deleted, then a T there was deleted, and a T there was deleted. Okay, so a biologist might prefer to have more contiguous regions. Okay, they don't want these hanging or these extra gaps. Okay, so I'm sorry, they don't want to see saying it wrong. Okay, they don't want to see gaps being split. They want to see gaps being more contiguous. Okay, does that make sense? I'm sorry if I confused you earlier. So this they will prefer because this comes from a single mutation event, presumably, and this one has multiple events that are causing that mutation. So one way to avoid these kind of alignments is to change your gap penalty and say that every time you put in a gap, okay, that's like a big event, so penalize. But whenever you're extending a gap, it's okay, give it a lesser penalty to do. So whenever you're introducing, so these guys will get a much higher penalty. That penalty will come only once here. These two are just extension of gaps, which will be smaller penalties, okay. So these are called affine gap penalties. So what you're saying is that there is a constant h, which is just extra, okay, which is given to every first gap. So the first gap is going to have h plus g penalty. All subsequent gaps are going to have an addition of g. Okay, so earlier we have had our gap penalty that looked like this. Okay, it was negative, but when I say penalty, I'm calling it positive. Okay, the score was negatively growing, right? Here it goes like this. Earlier it was going like this. So now what you're saying is introduce an h for every first penalty, first gap, and then let it grow. This is called an affine gap penalty. Okay, now this it is a little painful in the sense because it is not directly, now you cannot apply our earlier dynamic programming. Why? Because now it depends, right? If you're going up or left, whether you came there because of a gap, if you've already come there because of a gap, then the extension of a gap should be a smaller penalty. If you're introducing a new gap, it's a different penalty, right? So you can't directly apply the earlier dynamic programming approach. So what people do, or what Goto proposed, was that you have now 3 matrices instead of 1. So every matrix has a specific meaning. So matrix D will store the similarity associated with the alignment of the I prefix of X and the J prefix of Y, just like we had so far, but insisting on I and J to be aligned. Okay, so now you have an extra condition that you ensure that the ith and the jth letter are aligned. Okay, so that's like keeps the memory. It has so far been aligned. E now says that again it's the optimal alignment, but you insist on that on the top character, that means the X, should be aligned to a space. Okay, this one says that this is optimal alignment as long as you insist that the jth letter is aligned necessarily to a space. Okay, so now whenever you're extending, if you're extending from here, you know that it's because you've already gotten a gap. So now that gap can get a smaller penalty. If you're going to introduce a gap here from this matrix, then you give the extra penalty. So you can fill up your matrices like this. So because Dij is always insisting on the last character to be aligned, right? There's no option there. So you have to give that score, okay? And aligning a character does not penalize any, it's not like a new event. So you can extend either a gap to an alignment. You can take a gap and put a, okay, let me just write this down. So when you're filling the D up, right, if you're aligning the I and the jth character, it doesn't matter whether you're extending something like this or you have I minus 1, j minus 1 aligned and now you're doing an I and a j, or the reverse where you have an I minus 1 with a gap, j minus 1 here or anything here, doesn't matter. Right? These are the three possibilities, right? That you have a gap here, gap there, or you have an alignment earlier. And all three are treated equally, right? And you add to that and take the max. Okay? This one says that if you're putting a new gap into something that was already aligned, so now you're looking at I plus 1. If you're introducing a gap for the j plus 1 at 1, sorry, I plus 1 at 1, then you need to put that additional h. Again, if you had a gap in the other sequence, in this sequence if you're introducing a gap, then you have to put an h. So any introduction of a gap gives you h, an addition of h. Okay, and so you fill up these three matrices and take the maximum, and that helps you get the answer with an affine gap. Okay, so Dij is a special matrix, so it's not just stores the minimum, it stores the minimum maximum scoring alignment such that it insists on the I and the j to be aligned. So xi and xj, yj, align them. Then what is the best alignment? So it's a very special alignment. So it may not be the best one. Okay, this one says that the I, the j should be aligned. No, the I should be aligned with a space. It insists on that. Okay. And it doesn't matter whether the previous one was aligned to a space or not. Only that the ith one has to be aligned with a space for the y sequence. But yj cannot be there. That is the only thing. Yj has to be before space. We don't know what is happening to yj. And this one insists on an alignment such that the yj ends with a. But the alignment is specific, right? Alignment is between prefix up to I and prefix up to j. So that is there. So I is there somewhere, xi is there somewhere. It must be before, has to be before. In this case, yj is before and then there is a space. And here you're saying that I and j have to be aligned. We don't care about what happens to I minus 1, j minus 1. So when you're filling up these respective matrices, so here you're saying that I and j has to align, so that is that score, that alignment score. And because it's aligned, you just have to go diagonally up. And either one is, any of those three is okay, as long as it's the maximum. So it doesn't matter if I minus 1, j minus 1, both of them aligned with each other, or which is the first case, or whether the I minus 1 aligned with a space or whether j minus 1 aligned with a space. As long as x, j and yj align and you pick the maximum from the 3. Now this one is insisting on there to be a gap. Now this means if it takes a max from its own matrix, that means it's only extending. So an extension should not be penalized. That is why the h doesn't show up. In this case, you are starting a new gap, right? You are introducing a new gap, so you have to put the h in there. Here also you are starting a new gap, but it's an interesting gap because f just means that j was also aligned with a space and now I is also aligned with a space, but it's a new gap, right? Because the so far, so why is it a new gap? Is that clear? It's a new gap because the condition is like this: when you're extending the f, that means my j1 was aligning with a space up, right? So yj was aligning with a space and xi aligns with a space now. That is what your this means. If you pick this one, it means you are doing this, which means you're introducing a new gap necessarily because yj was there. So you are introducing it. So this is the memory kind of thing, right? So f is telling you what happened before. E is telling you what happened one step before. So it is just helping you keep that, whether it is a new gap or an old gap that you're extending. That's all it's doing. Okay, so let's, it's 4 now, so we can start with. Alright, here's again two different types of, two different solutions, okay, for the same problem. So you have two sequences. The first one is looking at introducing the gaps at the ends, and the second one again has two gaps, two chunks of gaps, but they are in between. Okay, so again, if you ask a biologist, the biologist will prefer to have something like that. Okay, because it just means that you had a smaller sequence to begin with. Why should you care about the edges, right? But even if you do the affine gap scoring, it will both score the same because the introduction of a gap happens 2 times, right, in both places. So that h is going to come up twice. So the scoring is the same. But if you ask a biologist for a meaningful alignment, she will prefer that one. Okay, so so in fact there is a gap there. There is an extra gap. So this one probably scores lower. I have not scored it, but no, turns out I have scored it. So this one scores a minus 18 and this one scores a minus 12. So this one is actually a better alignment using our scoring metric. This one is a worse alignment. But if you show this to a biologist, the biologist will prefer that one. Okay. So how do we handle this? So what this means is that you don't want to penalize the trailing gaps. Correct? So if you're scoring, you just stop scoring there. After that, forget about it. What does that mean in our matrix? We are filling the same matrix, right? The V matrix that we are filling. That means you can, or the top one, yeah, depending on where your Y is. So if you, so you can just take the max over here. Instead of going all the way there, just take the maximum element here and start tracing back because all these are gaps. So you don't have to care about them. Take the max alignment, score over here in this. So if this is lower, that's fine. You just take this, whichever one is a bigger one. And this works for the Y being on top, right? So symmetrically, you should also choose the max here. So whichever one is the best is going to give you, going to get rid of the trailing gaps, or is not going to penalize the trailing gaps. Is that clear? So you just take the maximum number here and here, and you start your trace back from there. Because then you just, what you're saying is just don't score anything that comes, any gap that comes after that. Okay? What do you do about the beginning? What about the starting gaps? You don't want to penalize your CAG matching with gaps. How do you fix this problem? How do you not penalize your first starting gaps? What do you do currently in your matrix? You initialize it to I times G, right? You are penalizing all those gaps. So what do you do to not penalize them? Put it as 0, right? So you just set these to 0. And then whenever you hit a 0, you know that everything else is a gap when you're tracing back. Okay, that way, so you're not doing anything new, right? You're constructing the same alignment, right, matrix, but then you are just looking at, so this is what I meant by many times when you're creating a model, as a mathematician or a computer scientist, you don't really know what makes sense at the end. So you start putting in assumptions that you think are reasonable, like the original things, right? That you have the very first alignment, that the exercise that you did. But then you notice that, you know, the affine gap penalty makes more sense. So you put that in. Then you realize that the hanging gap should be need not be scored. Put that in, and all this comes by, you know, talking or discussing or understanding a little more about the domain. Only then are you going to be able to formulate a more useful model. Okay. Of course, it not being useful is also, I mean, as a computer scientist, we'll have a lot of fun developing the algorithm for that. Okay, there is still merit in doing that, but if you want it to be used, you have to sort of be a little more careful and be open to the fact that your model may not be exactly what reality shows. And so to get there, you have to fix it as long as you keep your mathematical foundation sound. Okay, you don't do ad hoc tweaks, right? We've not done any ad hoc tweaks, right? Whatever we've done is we've added constraints, right, or situations or things, but they are all in a mathematical framework, right? So there's no ad hocness. Okay. So those algorithms are good when you want to align two sequences with each other completely. But many times what happens is you have two proteins which appear to do similar things, okay, but they're not identical. In which case your alignment probably a complete global alignment probably does not make sense, okay, because proteins have domains. You've seen that, right, in the previous lectures. The structure that they fold into, they have very specific domains which interact with others. So if you have two proteins that are doing very similar things, like they're receptors, right, presumably there's only one domain that is interacting with something else, right? Everything else is a little non-specific, in which case the kind of alignment you want to make is like this, to find out little pieces, as big as possible, but they can be small. Okay, so the official sort of definition would be a local alignment. So what we did so far was a global alignment. Okay, so local alignment of strings x and y is an alignment of a substring of x with a substring of y. It's clear what a substring is, right? Any little piece with no modifications. Where a substring must consist of contiguous characters. So we know global alignment, how to do that, right? We are pros now at that. By hand we can do. So so if we decided to do this in the brute force way, right? How many alignments would you have to do? So what brute force means in this case is take every substring of x and every substring of y. How many substrings of x are there? How would you define a substring? It will have, let's say, xi, xl to xk, right? From the original x1 to xm, right? So you need to define an L which is between 1 and m, and you need to define a k which is bigger than L, right? So now can you tell me how many substrings you can have? Your answer first. M plus mm, no, m. M. One length substrings will be m. Huh. Okay. Alright. What was your answer? Ha. So it's not really a power set, right? Because you cannot interchange. You can't have arbitrary sequences. It has to be contiguous. So I think your answer is fine. So what you need to do essentially are pick any two indices. So m choose 2, which is what this is, right? Any two indices you choose defines one substring, right? And there are m choose 2 ways of choosing two things. Does that make sense? No? Oh, look at the way how he counted it. He said that how many substrings of length 1? M. Agree? There is x1, x2, x3 all the way to xn. How many of length 2? x1, x2, x2, x3, x3, x4 all the way to xm minus 1 xm. So that is m minus 1. How many of 3? 1, 2, 3, 2, 3, 4, 3, 4, 5 all the way to m minus 2, m minus 1, m. Right? So that is m minus 2. And you keep doing this until you get your final one substring of length m. And this summation is m into m plus 1 by 2. Right? So m choose 2 plus a little more, yes. So m choose 2 would give a slightly different answer, but on the order of m square. Yes? And how many in y? In the order of n square, right? So total combinations that we need to compare are m square into n square, yeah? And each combination takes quadratic time, right? If you do global alignment, right? So why are we doing this? We want to find the best substring or the best alignment which is made up of a substring of x and a substring of y, right? The alignment criteria remains the same, that the score, right? That scoring is the same. You can have gaps and everything, okay? But this is the best alignment between any two substrings. So one brute force way of doing it is take every pair of substrings and do dynamic programming for global alignment. Okay, so that would then become ridiculously high, like m raised to 3 into n raised to 3, because it will be m square into n square into mn, which is the time complexity of global alignment. Of course, the sizes are going to be shorter, but still, it's going to be a huge complexity. Okay, so we want to do better than that. All right. So this algorithm says that, this is Smith-Waterman. Algorithm says that instead of storing, we normally stored the best alignment between prefix of x and prefix of y, right? So what you're saying now is let it store the best local alignment between the suffix of the prefix and the suffix of the prefix. Okay, so all it's saying is that this local alignment should contain xi and yj. Okay, whether they are aligned with each other doesn't matter, but they have to be there in the final alignment. But we may end up, when we go back up, we may end up here. We need not have the first part. Okay, so because if you look at the local alignment here, right? Sure. Right? This alignment does not go on to the beginning, so it will end somewhere. So this, so if you cut the thing here, so this is a prefix. Okay, so this is one prefix, this is another prefix, and this common thing is the suffix between the two of them. Does that make sense? No? Okay, let's go move on and see. So the way you fill up what this matrix is, did you understand what we are trying to find? We don't want a global alignment. We just want a little piece that does the best. Okay? We understand that if we decide to extend it further, we can get a negative score. We can go worse. So let us not go. Whenever we find a negative score, there is no reason to go on. Okay? So all we need to find is, in my prefix, let me find something that contains the suffix. Okay? That is my global alignment. So this is how you fill up the matrix. Okay? So this is the same as earlier, but you have an additional one, additional comparison, which says you just set it to 0 if your numbers become negative. Okay, so what is the minimum score in a local alignment? 0, right? When nothing aligns to nothing. So you should not get a negative scoring alignment. That cannot happen in a local alignment, right? So whenever the intuitive thing is that whenever you hit a negative number, you start again from 0. Say, I have not seen any of that. It is a terrible alignment till here. Let me start a new alignment here and start looking. And that's how you essentially fill up the matrix. Except that we said that the value of the optimal local alignment, this stores between the suffix of xi and yj, right? So where is the answer? Where is our final answer in this matrix? If we insist on choosing this region, we are insisting on using the last characters, right? So we need to, so where is our answer in this matrix? Earlier our answer was always here or there, depending on the problem we were solving. Huh? Yes. But which element? Which row? Which column? Huh? So everything is non-zero or 0. Right? So what are you saying again? Randomly. No. Now there is no brute force. We are filling it up in a structured manner like this. Right? We are saying that we are going to extend an alignment in the same way that we were doing earlier. But if ever we encounter, because of gaps or whatever, any of these numbers become negative, just set it to 0. So don't allow anything to be negative. So you will have lots of zeros in your matrix and there will be other positive numbers. But the answer may not be at the bottom corner. So where is the answer going to be?
Is this algorithm trying to push all the gaps to the
Not necessarily. Not necessarily. It could, but not necessarily. You could have the answer somewhere else also. So here it is not looking for gaps particularly. You might get a gap in between, that is fine. But once you get a negative, become really bad, becomes less than 0, you are just forgetting about it and setting it to 0. So you're forgetting that there was a gap. There is no recollection of how you got to that 0. Right? So here you know where you came from, diagonal. Here you know you came from top. Here you know you came from bottom. Here you just, that's it, you stop there. You don't go further.
Yes, because I can make these sufficiently less so that it will always become a negative. So that
Sufficient.
the gaps will be always to the right side of the string or in one side of the string. That is
That can happen.
because
But then it will.
penalty is very large, very small. Then
No, that if you put penalty very big, there's no. it doesn't mean
It will be
negatively very big, right? Big penalty. No. So then,
it will be either the
so it will, no, so what it will start doing is try to do mismatches. Since you're saying don't do a gap, it's trying to going to start putting mismatches and put gaps only when necessary. That is what it will do when you change the gap penalty.
because in the algorithm when you use that age
Yeah.
for one each extra gap.
Yes.
So it was kind of like it was pushing to go diagonally. Because
Not necessarily. It is
penalty is
no.
minus h minus d.
Correct. But once you start a gap, the G is actually small. I did not say that. The G is small, the H is big. So H is -2 and the G is -1 typically. So extension is okay. It's only the introduction that you're penalizing. So here what you're saying is that there can be any piece anywhere in your matrix. Okay, so because it could be the prefix of any suffix. That could be the answer. Right? It could be the prefix of any suffix over here and the prefix of any suffix over there could be the answer. And they're all stored in this matrix. So where is it going to be in the matrix? Yes? Just 1 second. He raised hand first. Yes. Yes. Yes, correct. So you could have multiple substrings, yes. Scoring the same, scoring. No, finally you are just finding one local little piece. If you have two such pieces, you will find two such pieces. If combining both those pieces gives a better answer, then that is the whole thing is the local alignment. Okay? So where is the, you were saying something. Why? Correct. But see, there is no diagonal here. The sizes are different. See, every I comma j in this thing stores the value of the optimal local alignment between some suffix of I and some suffix of j. You want to find the best one. So what should you do? Highest. So you just look for the biggest number in the matrix and that is your solution. If it is here, then you choose that one and you keep going back. How far back will you go? Until you hit a 0 from where there is no arrow coming out. Okay? So looking at your faces, I don't think you want to do this right now. So let me show you the answer. So this is what you get. Okay? So sure. So here, do you have your earlier matrix? It should be fairly similar. So first of all, initialization. This is going to be all 0. Just like the gaps earlier, right? You're not going to score a gap at all. The beginning and the trailing gap. So, sorry, beginning gap. This is going to be 0. That is going to be 0. Okay. Now, your first row will probably remain the same. So, let's go to that thing and try to figure out what's going on. It's the same. Right. So, this was the matrix, right? So, all these will be 0. Okay. So, you can't do it that way because the next numbers will depend on the previous ones. Okay. So, let's just do it. Let's just do a few numbers. Okay. Okay. So these zeros are okay. So when you're doing this one, right, you are either extending this guy and matching A and C, which is a negative number, right? Extending this one with a gap, which is a negative number, again a negative number. So it's a 0. Yes. So how would you normally fill this 0 plus minus 1, right? This is the matching is minus 1. 0 plus minus 2, 0 plus minus 2. They're all negative numbers. So 0. Okay, and I have not shown arrows here, but there is no arrow here. Okay, because you just came because all numbers are negative. Same thing will happen for these guys. Okay, they're all negative numbers. Now what happens here? So you again is negative numbers because T and A are not matched. Right? Now here you get some interesting stuff, right? Because you have a 0 plus 1, which is positive, so it gets the 1 with an arrow. What happens here? It is 0 plus minus 1 because the A and the T don't match. 0 plus minus 2, which is again negative, and 1 plus minus 2, which is again negative, so it's 0. This one is again 0 because it's surrounded by 0 and it does not match with T. So that's how you keep filling up. Let's look at the interesting one here, where you get a 1 plus 1 or a 0 minus 2 or 0 minus 2, right? This is again extending. Okay, so that, but you can get a gap. Okay, in this particular case, you don't seem to get a gap in your final answer, but you can get a gap. You can imagine there being a, after 3 you can get a gap and become 1 and keep on going up after that. Okay, so now where is the answer? There are two solutions. You either go from here or you count from here. Okay, in either case it looks like TAG. You stop at the 0. And if you just look at the sequences, that seems about, that makes sense, right? There's a TAG here that matches with here. There's again a TAG there that matches with there. So that both the two local alignments in this case are identical. Okay, so this is again just a tweak of the global alignment, which is actually used more often. Okay, so the reason I managed to make this graph is this chart is because I cheated, and there is a very nice resource. Okay, if you have your computers, you can try it out. Otherwise, I'll just show you on mine, which allows you to try out all these dynamic programming things with different okay, so this is mostly RNA, here is sequence alignment. So you can go to Needleman Wunsch. Okay. You can put in your sequences. You can put in whatever matches, mismatches, gaps you want. Okay, this is, it also tells you what formula it's using, which is the same as what we discussed. And you can then select results. So this is the output. In this case, there's a single output. Can you dictate the earlier program thing to me? What we were doing, ATAG1? I'll just type that in. Okay, I got it. Right. CTAGTAG. C now. See, it's auto completing because I got these numbers from there. Yeah. Okay. And it's telling you the two results. There's one. Here's the other one. Okay. This is actually something that you can also code and do, but this is a nice interface. Okay. All right. What else? This one is the one with the linear memory. Okay, with the divide and conquer, where I think I lost some of you. Where you align the m by 2 with a j or a gap. Okay. This one is another. This one is with the affine gap. So you can go there. You can choose what you want for the h and what you want for the g. Okay, so this is for the gap opening, this is to enlarge. They use a slightly different scoring matrix, scoring formula. It's very similar, but they don't use different D, E, F. They use an F, E, F, yes, but they sort of make do with a one lesser matrix, but the concept is the same. Okay. This one is the local alignment one. And this one can again, there it is. See, this is where I got the matrix. Okay, so you can try it out if you're interested in programming. It's a very nice exercise. You can write your own program and check with this. And whatever exactly what they've done, except for maybe the visual thing is probably much nicer than what you can do at this point, but everything else is very much doable. Okay, and maybe also the pointer trace back at this point you can skip, but definitely creating the matrices is something you can do. Okay, so the other thing I wanted to talk about was BLAST. Okay, so normally, so this is the basics, okay, of alignments, all types of alignments. But in reality, in practice rather, people don't use these because they are still very memory and time consuming. Because if you look at your, let's say right now you're just doing 2 alignments, okay, that is still just 2 sequences, that's still manageable. But let's say you want to choose from your database of all the current proteins, which one it is the best aligned to. You have to do this for all the proteins in there, right, in your database. So that becomes a little cumbersome to do it exactly. This is the exact method. Okay, all these methods were exact. There was no approximation. It was there was no ad hoc anything, right? That is how we define it, how we solve it. And so what BLAST does is they do a bit of a heuristic approach. So they know already what database they have, right? So they can do manipulations to their database in a way so that whenever you have a new sequence that you want to, whenever you give a new sequence to check with, they can do that quicker. So for them, it is not 2 arbitrary sequences. For them, it is 1 arbitrary sequence that you are feeding it and a whole set of sequences that they know very well. Okay, so the way they pre-process the data is they look at little k-mers, okay, beforehand. Okay, you know what a k-mer is? A string of length k. Okay, so it comes from polymer. Okay, so k-mer. And you just, and then they create some sort of a hash table, remembering where all the these k-mers are present. So instead of testing the whole gene sequence with all of them, they first do a quick search, okay, with these k-mers, and then they start, whichever k-mer works, they start extending the alignments, and that's how they do it. Okay, so you can go there as well. Okay. So the resolution is not how I would have liked it. So there are different types of blasts. You can do a nucleotide to nucleotide, protein to translate a nucleotide to protein. So you can say, I'm giving a nucleotide sequence which I know is a protein, and so on. So if you just put a nucleotide blast in there, just 1 second, see it better here. Okay. Okay, it says that you can enter your sequence. So this is a sequence I just right now downloaded from the browser. Okay, a big sequence that I've put. Let's see if it works. I've not tried this out. You can see that it is not auto-completing. Okay, and I keep everything the same. So what does it say? What do you want to test? Do you want to test all non-repetitive regions, or and the nucleotide only, because this is DNA, right? So we only care about DNA. So you can do all these, you can select whatever you want and say blast, and it keeps doing something. It's a very, very popular resource, and they have many machines in NCBI. That's who hosts this, right? That's the institute that hosts BLAST, and lots of I think they have an online map as to who is using it right now, that kind of stuff, and there's lots of people using it at a time. Okay, so it said that it matches something. Okay, this is sorry, I don't know how to fix this resolution so that you can see. It gives this maximum score, and here it is aligning, giving you the alignment. Okay, so our range was from 1 to 5,000. What does this mean? Wait. So our length was, so it found that it matches the GATA binding protein, which is the one I took, by the way. So I took the GATA binding protein from the NCBI, from the genome browser. You remember the genome browser I showed you last time? So I just copied that and I pasted it in that box, and it detected it, okay, from its big set of lots of different sequences, different databases that they have combined. And it's pretty much an exact match. It's saying the number of gaps is 0, right? Number of identities is complete, and so on. Okay, so it's doing whatever it's supposed to do. Does not mean it will always do the correct thing, because it is not provably optimal. So it is a bit like the greedy things that we have seen before. It tries to do its best, but there's no guarantee that it's going to do the best each time. Is that clear? Our methods will do the best each time, but they will take a long time to run. And if you're doing it on paper, it will take an enormous amount of time. Huh?
Can anyone use this?
Yes. No, it's all free. It's all free. You can.
If I put any sequence there,
Yes.
so you will tell me that if it matches with any other registered sequence.
Yes. And there are many registered sequences. So you can choose. If you just want to focus on human, you can just focus on human. If you just want to focus on bacteria, just choose the bacteria ones. And you can, it has different methods. If you give it a very short sequence, it has a different method of calculating. It has a few different heuristics that it uses each, depending on the question that you're asking. Okay, if you have a protein, also it will do it differently. Yes?
So any other way to know that you said that you have taken that particular sequence? Okay. So is there any other reference that where you can find this kind of sequences? And
Yes. So Santa Cruz Genome Browser, the one that I showed earlier, that website also has lots of genomes. So you can try giving it, you can download a few, make a few changes if you want, mutate a few things, and then send it and see what you get. Okay. Because presumably whatever is on the Santa Cruz Genome Browser is here as in its database. It must be.
Is that this long language?
No. That was just the person. UCSC Genome Browser. Yes. You can select whichever organism you're interested in. Okay. Okay, I'm losing charge. It's not good. Okay, let's go back to a little bit of lecturing, and then we'll stop. Okay, so now we're going to start. Keep the discreteness a little behind, and talk about probabilistic models. Okay, so the big question that we're trying to solve is, now that we know the human genome, we know many other genomes. So we have this, lot of sequence. right? We want to know what it means, right? So how can we figure out what these different regions mean? Whether there's any point in learning identifying what they do. Okay, so there's some things that we've already done, right? Align to things that we already know, which is what BLAST does, right? So if you have a new sequence, align it, send it to BLAST, and check which sequence it closely resembles. The other thing you can do is, see if there are any specific patterns in the sequence. Some sequences are very AT rich, which means they're filled with A's and T's. Some sequences are more GC rich. Okay, so some organisms are very inherently, they have a lot more AT than CG, and whether that means anything, right? So ideally, I would like to use a good spelling for that, but we would like to annotate regions. Okay, so we want to annotate amongst the whole genome what is doing what. Okay, so if you have your whole book of recipes which you cannot read, you want to be able to tell that this is a recipe for this protein, right, for this meal, and it is executed because there is this other recipe that is telling it to execute it, and so on. Okay, so you want to know that. So you have the you pretty much have the hardware, right? You know ACGT, everything. You just don't understand the software that is running it. You don't know where it is, what is doing what. Okay, so we need to first understand a little bit about what a gene structure is. Okay, how many of you have taken a basic genomics class? So you know this. You know this. Anybody else who knows, who has seen such pictures before? So good. So earlier the first lecture I showed a gene which was one contiguous piece, right, in green or something. So in us, in eukaryote, in more complex organisms like us, we don't have one necessarily a contiguous piece of recipe. Okay, so our recipe for a gene, our gene is sometimes split. Okay, so when the RNA polymerase needs to make this gene, right, this arrow is telling the direction, okay, of reading the gene, because you remember that we have two strands, so you can read it from here or from there, but the gene makes sense only in one direction because the starting codon is an ATG, which should come on one of the strands, right? So the way our system works is that your RNA polymerase comes and does whatever it's doing in bacteria, which have this contiguous genes, starts copying. Okay, and when it copies this whole thing, there is another enzyme that comes and cuts out the redundant or the unnecessary pieces. These are called introns and these are called exons. It's okay if you forget of these names. Okay, I will introduce them again later on when we are talking about the models. Okay, but what happens is that when the RNA polymerase creates the protein, creates the RNA, it is creating it only based on the genic regions. It is cutting out and joining the remaining pieces together. Splicing, they splice wires together. You know what that means? So it's like that. So you cut and then splice. Okay. There are other enzymes which do it. So one question you should ask is why, right? Why do you need to do this cutting business? Why can you not just create? So you'll notice that this length is equal to this plus this plus this. So is everybody clear about the splicing thing? How it happens? Yes? Or should I just repeat again? Repeat? So you have your DNA, right, which is double stranded. That DNA is opened up and the RNA polymerase starts reading the one strand that it's supposed to read. And then there's another enzyme that comes and figures out that these pieces, okay, should not be there in your final thing that goes to the ribosome for translation. And so it comes and cuts over here and cuts over there very specifically, right? It does not cut in between. It knows where to cut. And another enzyme comes and connects these two. and then this little piece of RNA is destroyed. Okay, so there's a lot of unnecessary stuff going on. Again, this little piece of RNA is destroyed. It's just cut out. So the reason we have this is because it adds a layer of complexity. The enzyme can decide which ones to skip and which ones to keep. Okay, so it might decide to keep 1, 2 and 3 exons which are in this color. Okay, or it might say, you know what, I also don't want this one, let me just keep this one and that one and create a smaller one. Okay, so it's the same gene, okay, but can have variants. Okay, this is where combinatorics comes in. So there are many options, right? It might decide to chop these off along with this, or it might say that I don't want the first part itself and I'll start, maybe I have a transcription start site from here and have a smaller one. And what this does is gives you, the same gene gives you different types of proteins, because now your certain domains are just missing, right, or added, and it gives you that ability to control from just a single gene. Okay, so how does this happen? How, both things, right? How does the cell know that this gene should be expressed? How does the cell know that this exon, this little piece, should be spliced out or should be there? Okay, so there are many different signals around the gene, or sometimes far away from gene, but in close proximity, so the DNA turns around. Okay, these are called little pieces, signals, which are bound by different proteins. Okay, so the protein finds its partner and then decides, you know, brings about some conformational changes in the DNA, promotes transcription, or sometimes hinders transcription, and so on. Okay, so this time it's promoting. Okay, so that is just one type of, these regions are called regulatory regions because they are regulating the gene. They are themselves not genes. Okay, this is really tiny, this piece of region, and this is to scale. Okay, so this is to scale, as in a TF does bind a really tiny region. Okay, so but your regulatory regions can be really tiny. Your gene is typically thousands of bases long. Okay, so this means that our genome has some structure. Okay, so you have a gene, you have these regulatory regions which are controlling. So can we now build models that will recognize DNA sequences of a certain type? So given a new sequence, can we say, given a new unseen genome, can we say, okay, this, just looking at the genome, this looks like a gene, these are the exons, this might be spliced out, this looks like a regulatory region. Okay, so we're going to talk about probabilistic models, okay, to do this. So I'm going to assume that you guys know probability, right? Anybody who doesn't, is not comfortable with probability, sample space, events, probability of some event, you know, yes? No? I don't understand that nod. Okay. So who's expert in probability? They can explain. What is probability? The people who confidently said they know can explain to the ones who are not confident. We actually did an example of probability once when we were looking at the terminator thing. So we said that there was a probability of getting a terminator nucleotide and 1 minus that probability of getting a non-terminator nucleotide, or the reverse, whatever it is. So that was a distribution, okay, that was a probability distribution over two possibilities, okay, whether it's a terminator or not, like a coin toss. Okay, so a coin toss is a probability distribution over head or tail. Okay, so you can, if it is a fair coin, then the probability of a head is half, probability of a tail is half, right? Okay. That's about to begin with, this much probability is okay. Okay. Sometimes you can have continuous probability distributions. This one was a discrete one because there were only two possibilities, head or tail, right? You can have a normal distribution where you have a range which is continuous. Okay. Conditional probability. No? Yes? Okay, let's go over that. I think people are I'm not getting any sense of, do you guys know conditional probability? Yes? Can people raise hands if they know conditional probability? I will not ask them to explain. Can you please raise your hands? One person only. High up now, come on, I can't see. Okay, yes? No. Okay. Yes, no. Alright. So when we talk about probability, we generally have some sample space. Okay. Let's say we have two events that can occur. Okay. Let's say you have an event A and an event B. Okay, that may or may not occur. So event A could be that it rains. Event B could be that you are carrying your umbrella. Okay. So a conditional probability means something like this. What is the probability that it rains given that you're carrying your umbrella? Okay. So you have a probability of this. You have a probability, you sometimes take your umbrella, you sometimes don't, right? You have a probability of that. So you have P of A. You also know kind of what P of B is. So conditional probability means that given that you have taken your umbrella, what is the probability that it rains? So this is defined as the probability that it rains and intersection set theory. I'm assuming everybody knows Venn diagram sets. Otherwise, I give up. Okay, so A happens and B happens. Okay. And you need to normalize by all the times when B happens, okay? Because this is conditional on B. Okay. So many times instead of intersection we write comma, okay, which just means A happens and B happens. Okay, divided by the probability of B. That gives you the probability of whether when you're carrying your umbrella it rains or not. Okay. So if you count, you will count all the situations. Let's say you count 1,000 times that you carry your umbrella, 1,000 times, okay, that it rains. Count the number of times both these things happened. You will get that answer. Okay. That's called the conditional probability. Now, if you carrying your umbrella, if it's raining, if it is the probability that it rains, given that that you're carrying your umbrella, is let me put it differently. If you don't look at the weather forecast at all, and you know nothing about the Chennai weather beforehand, that there is a receding monsoon or anything, you don't know any of that before, you don't know you assume nothing, no knowledge, then technically the probability of it raining and the probability of you taking your umbrella are independent. I mean, it raining and then you taking your umbrella are independent events, right? So if that happens, then the probability of A and B happening actually is equal to probability that A happens and B happens. When you have independent events and both are happening, it's just a product of them divided by probability of B, which means this is happening. Okay, which means the probability that A happens given that B happens is the same as whether the probability A happens, because it is independent of B, gives no. Okay, So it is, so this is essentially a little bit about conditional probability. The reason I'm talking about conditional probability is because we are going to use a little bit of Bayes rule, okay, which comes one step after conditional probability. Yeah. So probability of A given B is the probability of A into probability of B given A divided by probability of B. Why? Why does this happen? We can do it by first principles, right? What did we say P of A comma B given B was? We said it was P of A happening and B happening, right? Divided by the probability of B happening, right? So probability A happening and B happening is also equal to probability that B happens given A into probability of A, because this intersection is commutative, right? It doesn't matter. A happening and B happening is equal to B happens given A into P of A. Okay, it's just conditional probability written a different way. You get that expression. Okay, this is also called the Bayes rule. Okay, because many times you know these numbers, but you don't know that number or that value. Okay, so for example, this is what I normally give for explaining Bayes theorem. Okay, so let A be the event that you have a biased coin. So wait, hold on. So consider an experiment, okay, where a fair coin and a biased coin are there, okay, and we choose a coin and toss it twice. Okay, so you have one fair coin and one biased coin. I have no idea what the bias is, but hopefully this describes. Okay, you have one fair coin and a one biased coin. Fair coin means 50-50, chance of a head. You choose a coin, toss it twice, okay, you get heads both times. And I think the bias is that it is always heads. Okay, so the biased coin always gives you head. The fair coin gives 0.5 probability head and 0.5 probability tail. Okay. Okay, so you chose one of these, you don't know which one it is, and you tossed it twice. Okay. Now you got heads, two heads. You chose the coin, tossed it twice, you got two heads. Now you want to find out what is the probability that you chose a biased coin. Did you understand the problem? Let me define it in A and B. So A is the event that you did choose a biased coin. Okay, and B is the event that you got two heads. What do you want to find? The probability of A given B, right? You've now been told that you got two heads, so conditional on that, what is the probability that the coin that I picked was biased? Right? Somebody's telling me the answer. What is the answer? 0.8? Okay. Alright. Anybody else? Let's go through the process. Okay. So what is P of A? What is the probability that a biased coin was chosen? Half, right? Because you chose it at random. So this is half. What is the probability that you get two heads given that the coin is biased? 1, right? Now, what is the probability that you get 2 heads? How do you solve that? So you can split this P of B by doing the following in this format. So P of B is equal to P of B given A into P of A plus P of B given A bar into P of A bar. Correct? Okay, this process is called something, do you know? At least we used to call it marginalization. I don't know what it's called. Marginalization, right? So what this is saying is that you're trying to find the probability of some, this is your Venn diagram that you draw for probability. So your sample space. This is your probability of A. This probability of A you can compute by cutting it into pieces and summing up each piece, which is the same thing, right? So here you are saying that cut up my B into 2 possibilities where you have a B and an A bar and a B and an A. So A bar means that you chose the fair coin and A means that you chose the biased coin. Okay. So what is that? So 1/2 divided by B given A into A is again 1/2, right? What about the A bar? What is the probability that you get 2 heads given that you have a fair coin? Is there a mistake? No. Just tell me. Eventually you have to get 0.8 if he's right. So, what is it? 1/4. Okay. 1/4 probability of getting a head when you picked a fair coin times the probability that you picked a fair coin, right, into half, correct. And this is equal to 4/5. Yes, good. Okay, so this is how you use Bayes' rule. We will use a little more of it later. Is that clear how we calculated it? Okay, just for fun, try out this situation. Okay, that you have a swine flu test. Okay, which is accurate to a great degree, like 0.99, okay, or 99%, which means that if you have swine flu, okay, it's going to detect it correctly 0.99 times. Okay, if you don't have swine flu, and it will predict it as a non-swine flu also 0.99 times. Okay, so there are 2 errors, right? Both errors are equal. Is that clear? There is some error in the test. Both of it is 1%. Okay. Now let's say you go to a doctor, and the doctor says, go get a swine flu, or maybe you go to the doctor with a swine flu test done, and say that it's come up positive. Okay. What do you think the doctor should do if the doctor knows that about 1 in 1,000 people are generally affected by swine flu? Okay, so let me just put this in here. Okay, I'm not going to solve this. You can try it in the hostel. Okay, so swine flu test is 0.99 99% accurate, which means probability that you are detected as a yes given that you are infected is 0.99. Okay. Probability that it does not detect that you have swine flu, given that you are not infected. Sorry, probability that, let's say the other way around. Probability that it says that you are infected when you are not infected, 0.101. Okay, so the error is the same 0.01 in both places. Okay. The doctor knows, right? Doctor has some expert knowledge, right? Says that, just because you came up to me saying that you have tested, I don't believe it, because only 1 in 1,000 people with a cold symptom actually has swine flu, is infected. So what do you think is correct? What is the probability that you really are infected, given that your swine flu test comes out as positive? Is it clear? So you can translate 1 in 1,000 in terms of probability, right? So what this is saying is the probability that you're infected without knowing anything, right, totally, is 1 by 1,000. Repeat the question. So you went and took the swine flu test because you had a cold. Swine flu test came out as positive. The person administering the test says that there is an error rate of 1% in our test, and it's 1% in both directions. So the swine flu test will say that you are infected when you are not infected with a 0.01 chance, and it will say that you are this will say that you are not infected when you are actually infected, also with 0.01 chance. Okay, so that it tells you that these are the error rates. This is the best we can do. But the doctor looks at the result and says that I understand that you have come to me with a swine flu test, but my prior experience, so I have treated many people, I know that the probability, without knowing that you took the swine test or anything, is really tiny. It's only 1 in 1,000. Okay? So probability of I, I'm almost close to solving the problem here. So probability of I is 0.001. Okay? So you need to find out, after knowing all these facts, what is the probability that you really have swine flu, given that your test is yes? Yes, test is yes. That is why you're doing all this. Okay. All right. Do you guys have umbrellas? Yes, good. Since I'm going to be stuck in this building, I'm going to take as long as the rains takes to stop. Okay. All right. So a lot of many problems in biology are of inference. Okay, like you saw, right? You want to infer what a DNA sequence must be doing. Okay. So we need to have a sense of the different ways of inferring probabilistic models. Okay. So for example, let's say let's go back to the coin problem. Okay, and I tell you that I have this coin and I don't know whether it's biased or not. Can you help me find out? So if you look at it, you can't tell anything. Okay, just looking at it. But you can toss it and calculate the number of heads and the number of tails. Okay, so let's say you toss it n times. Okay, and you see k heads. Let's say you toss it 100 times. Okay, and you saw 70 heads. Okay, what do you think the bias is going to be? So I've actually come to you with a coin and I've asked you to find the bias. Okay, infer the bias of the coin. Okay, and you cannot take apart the coin. The only thing you can do is see the output, see the behavior. So the behavior says that out of 100 tosses, you saw 70 heads. Now you have to infer the bias of the coin. What do you think the bias is? Is it 50-50? What do you think it is? Just guess. No, it's okay.
The sampling, normally this 100 samples, the first 100 samples is maybe 70 days.
Yes.
So take another 100 samples.
No, you can. I don't. There is not enough space, time to do that. 100 samples, and you got 70 heads. What would you guess? Okay, let's say out of 100, since you're very hesitant, let's say out of 100 you got 90 heads. What will you say? It's biased, but what is the bias? So the next time I toss a coin, what is the probability I'll get a head? 0.9, right? So you just did some inference in your head, okay? So there is a more rigorous way of doing it, which is called maximum likelihood estimation. You've done this? Maximum likelihood? Do you know what likelihood is? Likelihood is the probability of seeing data given some assumptions about the parameters. Okay, so we have D, which we will typically use for data. Sometimes we also use X. Okay, I will interchange. Please excuse me. Just like I interchange min and max, I will do that. Okay, so this is my data. I will have a theta for the parameters. Okay, in this situation, for the coin toss, I have a single parameter P, which is the probability of a head. Right? That is my P. That's all that I need for the coin. And when I want to identify P, what you just told me, you just did this. You wanted to maximize over all theta this quantity. This quantity is called the likelihood. Okay. What is the probability of seeing 100 heads, 90 heads, okay, given theta? Okay, so what is the probability, let's make it more abstract, of seeing k heads from n throws or n tosses? What is this probability? Forget about a maximization, this function. You've seen this. It's a binomial, correct. So what is it? What is the answer? N choose k. Correct? Everybody knows, has seen this? Yes, okay. So this is your likelihood function. Right? This is the probability of seeing those k heads, 70, 90, whatever it is. So how do you maximize? So you want to maximize this function over theta. Derivative. This is literally the only time that you will see me do any calculus. Okay. So you take the derivative of this and equate that to 0. Right? Can you do it? Tell me what you get. You've forgotten how to take derivative of a product or you know the answer. Is this correct? Right? Okay, good. So now this you need to equate to 0. Something is wrong? Correct. I have one, correct. So I have 1 minus theta here and I have a theta here, right? K. Minus K. Thank you. So many things cancel out, right? This is correct? I can cancel out as long as theta is not 1 or 0. We will check those cases later. We should, in principle. What is theta? K by n. Yes, good. Okay. So you just did inference right now. You inferred using maximum likelihood. You maximize your likelihood and you got theta to be 70% or whatever, right? That is the K by n. Another way of doing this is a Bayesian approach, which says that sometimes the data is not enough. Okay? So if I just toss the coin 4 times and I get 3 heads, okay, I really don't think that the coin is biased to 75%. Would you think that? Let's say you tossed it once and you got a head, right? So it kind of your confidence kind of goes down, right, when your number of trials are less. So what a Bayesian says is that I'm going to use some of my prior knowledge, because not most coins are fair. Okay, biased coins are very rare, right? Unless you are kind of working in Cholet or something, right? It is a very rare occurrence to have a biased coin. So I'm going to just add a few unseen observations of mine. Okay, so Bayesian will say that I'm just going to assume that I tossed the coin twice and I got 1 head and 1 tail. And now tell me, what do you got? And I will add it to that. Okay, so what a Bayesian will do is say, do the same calculation, except that he or she will add something to n. He'll add 2 to n, add 1 to k. If you want to add a fair, if your bias is fair. Okay, so this would be k plus 1 by n plus 2. If you do this, it's called not maximum likelihood. What is it called? Has anybody done this? Anything? Anybody? So what we did, what we maximized earlier was probability of data given theta. Now you are saying that you have some prior knowledge about your theta, right? So instead of updating your belief about the data, you are actually updating your belief about the theta. Okay, so what you are calculating really is the probability of theta given that I have now seen all this data that you're telling me. This is equal to the probability of, just using Bayes' rule, correct, divided by P of D, which is a bit painful. Okay, P of D is a little annoying, but we don't want to calculate this quantity, right? We want to calculate the maximum, or maximize this. So when you're maximizing this, don't care about the P of D, right? It is independent of theta. Right? Technically, this denominator is that earlier denominator of that getting heads, right? The probability of B that we got, and we split, right, into all possible A and A bar. Technically, you should be doing this, P of D given theta and so on. Okay, but we don't care about all that, because P of D is independent of, it does not have a theta term in it. Okay, so this becomes the same as maximizing this quantity. And if you're a Bayesian that thinks like this, that you should just add a few counts, what you're saying really is, I have a prior over my theta, which is of a very specific form. It is like a beta distribution, okay? Anybody knows what a beta distribution is? Doesn't matter, okay? I can tell you. So this is the beta distribution, which says that it is proportional to theta raised to alpha and 1 minus theta raised to alpha 1, alpha 2. Okay, so this you are multiplying. It's proportional. There's some crazy term that normalizes with some gammas and things. Okay, that's why the proportionality. These are your hyperparameters, which are your 1 and 2. Okay, so what happens when you add this or multiply this quantity to your likelihood? Your likelihood anyway looked like this, no? It had theta raised to k and 1 minus theta raised to n minus k. Very similar looking, to which you added your prior. Okay, which looks just like very similar to that. In fact, the final product looks a lot like your prior. It's a very special prior that makes sure that even after seeing the data, your belief structure remains the same. Your distribution of your belief does not, only the parameter changes. So this alpha is essentially 1 and 1. It is actually minus 1, but doesn't matter. Does that make sense? So what you're instead maximizing is your belief of your theta, of your parameters given t. This is another way of doing inference, especially when you have little data. So what happens when you do get a lot of data? So first you were a little hesitant to do a 3 by 4, right? So instead you did a 3 plus 1 by 4 plus 2, which is half, no, 4 by 6, 2 by 3, right? So from 3 by 4, you went to 2 by 3, okay? Now, if somebody tells you that, well, I did this 300 times, 400 times, and I got 300 heads, you're still adding a very small quantity, right? This is now going to approach 3 by 4. Okay, so as your data increases, right, as long as your prior is sensible, your prior, your alpha and alpha 1, alpha 2 could have been off the charts, right? You could say that I have tossed the coin 1,000 times and I have seen 500 heads and 500 tails. You could always say that if you wanted to, but then you have to make that clear when you write the paper, what your assumptions were, what your hyperparameters were. And many times Bayesian methods work especially for biology because you can use expert knowledge in the model itself in a sound framework. So you're not doing any kach-pach in your, so your it has to be mathematically sound. Alright, I think we'll stop here. Thank you for your patience. We will do more of this probabilistic models tomorrow. Yes. Yes.